Thanks so much for the introduction and for the opportunity to speak to you today. So I'd like to first acknowledge that the traditional owners on the lands on which we're all meeting today, which are the Larrakia people here in Darwin, and I'd like to pay my respects to their elders past, present and emerging. So my talk will be in two parts today. The first bit will be on the applications of genomic sequencing for public health control of TB sort of generally. And then in the second part of the talk, I'll talk about my PhD work, which is on the genomic epidemiology of TB in the Northern Territory top end. No, it's not, there we are. So in terms of the Mycobacterium tuberculosis genome, it has a relatively low mutation rate with no clear evidence of horizontal gene transfer, which means it has less genetic diversity than many other bacterial species. In terms of the laboratory workflow and potential applications for TB genomic sequencing for public health, so the lab receives a sample, usually a sputum sample, but not always. It undergoes culture and then sequencing, often on an Illumina platform, which then generates short read sequence data. These get aligned to a reference genome, and because of the lack of diversity within the TB complex, the reference genome that's almost always used is that of H37RV. And by aligning to the reference, variants are called, and a list of SNPs or single nucleotide polymorphisms is generated. And these are then used to predict drug resistance, to type the strain, so to determine which lineage and or subspecies within the M. tuberculosis complex it is, and for cluster analyses. And all of this information can then be put together in a report which can be provided to public health and treating clinicians. So in terms of AMR prediction, there's been a lot of recent work by the Cryptic Consortium, which is run out of the University of Oxford and is funded by the Wellcome Trust and the Bill and Melinda Gates Foundation. Their ultimate goal is to achieve sufficiently accurate genetic prediction of resistance to most anti-tuberculosis drugs so that whole genome sequencing can replace culture-based DST, drug susceptibility testing for TB. And they've recently posted quite a number of manuscripts on various preprint servers based on their recent work. So recently they've looked at over 15,000 isolates, which have been sequenced and have undergone phenotypic drug susceptibility testing with broth microdilution. They've used these data to determine the epidemiologic cutoffs for phenotypic drug susceptibility testing. They've done genome-wide association studies to identify mutations associated with antimicrobial resistance. They've developed a machine learning model for resistance prediction. And probably most importantly, they've added all this additional data, to existing data to produce the WHO catalogue of mutations in Mycobacterium tuberculosis and their association with drug resistance. So this catalogue was published early this year. It includes only high quality phenotypic DST and whole genome sequencing data and has been analysed with a standardised pipeline. And this catalogue includes over 1,000 mutations associated with resistance and provides an estimate of confidence for each of these mutations. So it's a really massive and impressive effort, all of that work. So back to the SNPs, once you've sequenced your genome, these can also be used to determine both the subspecies within the M. tuberculosis complex and the lineage within that complex. And there's a tool called Snippet that can be used to do this. There's also another catalogue of SNPs that can be used to define sublineages with the Mycobacterium tuberculosis complex. And that's implemented in a tool called TB Profiler. So they're both very helpful. For finer scale cluster analyses, so actually looking at the way your genomes are related to each other and whether they're potentially linked or not, the usual approach is to calculate the pairwise SNP distances, and then to use these pairwise SNP distances for hierarchical single linkage clustering. Various studies have investigated the SNP distance thresholds that are appropriate to use when doing this, and these are two charts from a 2013 study where they looked at the SNP distances between isolates within the host, so at different body sites and longitudinally collected isolates over time, and also between epidemiologically linked isolate pairs. And based on this fairly early work, they suggested that a SNP distance of less than or equal to 5 SNPs was appropriate, was suggestive of probable transmission, and less than or equal to 12 SNPs was suggestive of possible transmission. And really since that time, these kinds of thresholds have largely held up. So core genome MLST is another approach. There is a core genome MLST scheme which has been devised and published. This is based on, so the scheme for Mycobacterium tuberculosis is based on 2,891 genes, which are shared by almost all of the Mycobacterium tuberculosis complex strains. And these can be analysed by comparing by determining the pairwise allelic distance. The authors of this paper actually used the same data set as I showed in the previous slide, where they were validating the SNP distance thresholds. And you can see that the pairwise allelic distances sort of follow a very similar distribution. So all of this information can then be combined into a report. So that might include the lineage and sublineage of the strain, a list of any resistance associated mutations and their interpretation, whether or not it was part of a cluster. In this case, it actually includes a phylogenetic tree, which shows where the patient's isolate fits in the context of other isolates that are part of a cluster. So in some instances, this might be included altogether in a report, or it may be appropriate in some instances to provide the drug susceptibility information to clinicians and the clustering information to public health. And different labs will probably have different approaches to this in different jurisdictions. So whole genome sequencing for public health for TB has been taken up fairly widely now. So Public Health England began prospective sequencing of TB isolates in 2017, and the US in 2018. In Australia, it's in progress with different jurisdictions at different stages. The Communicable Disease Genomics Network formed in 2015 with the aim of implementing genomics in clinical and public health microbiology practice, and has been an expert reference panel under the Public Health Laboratory Network since 2019. There is a TB working group within the CDGN, which is led by Ivan Bastian and Vitaly Sinchenko, and includes representatives from all jurisdictions, as well as the National Tuberculosis Advisory Committee. And the aims of this working group are to develop standardised approaches across Australia for genomic sequencing analysis and reporting. to help with development of a national analysis platform, which is the AusTracker platform, which has been developed by CDGN and is in use for COVID-19, and to assist with capacity building across jurisdictions. So this is sort of what's coming. So moving on to the next part of my talk, which is on the genomic epidemiology of TB in the top end. So the Northern Territory, a bit of background about that, it's got a population of around 250,000 people, but is very sparsely populated with an area of 1.4 million square kilometres. It's culturally very diverse. So a third of the population are Aboriginal people, around 3/4 of whom live in remote regions, and 20% of the population were born overseas. So our study was set in the northernmost part of the Northern Territory, what we call the top end, which is much closer to Indonesia and Timor-Leste than it is to other major Australian cities. So our study included all culture-confirmed cases of TB in the Northern Territory top end between 1989 and 2019, and we combined patient epidemiology with genomic sequencing of Mycobacterium and tuberculosis isolates to identify public health priorities for TB control in our setting. So over the course of the study, there were 741 cases of culture-confirmed TB. 329 of those were in First Nations people, 328 were Aboriginal people, and there was one Torres Strait Islander person, and 359 were born overseas. Of those who were born overseas, 2/3 of them were from Southeast Asia, with most cases from Indonesia, Timor-Leste, the Philippines or Vietnam. Among Aboriginal cases, there was a greater proportion of child cases. The majority of Aboriginal cases were living outside urban Darwin. There were no Aboriginal cases with HIV infection or any Aboriginal cases with multi-drug resistant isolates. 12.5% of First Nations cases died prior to TB treatment completion, which was much higher than among those born overseas. In addition to that, the median age of those who died was lower in First Nations cases, with a median of 49 years and a range of 21 to 79 years, than in Australian-born non-Indigenous cases, where the median age was 63 years, or in overseas-born cases where the median age was 77 years. So Aboriginal people had a high mortality rate, and the age of those who died was significantly younger than in the other groups. So the number of cases in First Nations people dropped significantly over the 30-year period. The number of overseas-born cases fluctuated depending on regional policies and politics. So, for example, there was a spike in cases in 1999 associated with the evacuation of around 2,000 people from Timor-Leste during the conflict associated with their transition to independence. And there have been increases in cases notified in the Northern Territory during periods of onshore detention of people arriving by boat. So whether that includes asylum seekers and crew, and Indonesian fishers who are fishing illegally in Australian waters. So likewise, the TB incidence in First Nations people dropped markedly over the course of the study, on average by 5% per year. However, in 2019, the incidence was still 15 per 100,000 population, which is significantly higher than the Australian-born non-Indigenous population. I think nationally, the incidence in Australian-born non-Aboriginal people is less than 1 per 100,000. So there's still a significant difference there. So we were able to sequence genomes from 2/3 of the culture-confirmed cases over the 30 years of the study. This is a phylogenetic tree, including these 497 sequences from the top end. Lineage 4 was most common. This is a lineage that is based on sort of phylogenetics, is predicted to have arisen in Europe with global spread associated with colonisation of the Americas, Africa, and the Asia Pacific region. And lineage 1 was next most common, and this is found mostly around the rim of the Indian Ocean, including in Southeast Asia, including Indonesia. So 93 cases had documented previous contact with a pulmonary TB case belonging to the same sublineage. And in 85 of 93 of these instances, the isolate pairs were separated by less than or equal to 12 SNPs and a median of 2 SNPs. So that's consistent with previous studies. In the final 5 study years, and this was the period for which contact tracing data were most complete, and is also sort of most interest to us because it's what's happening now as opposed to 30 years ago. There were 10 genomically linked case contact pairs, and of these, the contacts had been diagnosed between 4.5 months and 24 years earlier, the median of 6 years. So although this is small, these are small numbers, it does suggest that both reactivation from latency and recent transmission are contributing to incident TB in the top end at the moment. So based on a threshold of less than or equal to 12 SNPs, we defined 28 genomic clusters, including 250 cases, with 11 of these clusters having more than 5 cases, or 5 or more cases. Of the clustered cases, the vast majority were Aboriginal people, so 86%. 76% were from a remote region outside urban Darwin, with the Roper Gulf region and West Arnhem being most affected. The genomic clusters with an overseas-born case as the first case were small, and there were very few of these. These included between 2 and 5 cases, and no drug-resistant cases clustered together, suggesting that we didn't have evidence of transmission of drug-resistant TB in our study. Of 19 clustered cases in the final 5 study years, 12 belonged to the same cluster as a known previous contact. So the genomics supported the epidemiology. 1 belonged to a different cluster to their contact, and 6 had not been identified as part of any previous contact tracing exercise, suggesting a role for genomics in addition to epidemiology in understanding TB transmission in our setting. So there were 7 cases that had a recurrent episode of culture-confirmed TB during the study, and we had isolate pairs for sequencing in 5 of these instances. These recurrent cases occurred between 1.2 and 19.3 years after initial diagnosis. All of them had received directly observed therapy, but 2 had been lost to follow-up during treatment. And none developed drug-resistant TB during their second episode, or had drug-resistant TB during their second episode. Of the 5 isolate pairs that were sequenced, in 4 of 5 of these cases, both the initial and the recurrent Mycobacterium tuberculosis genomes belonged to genomic clusters. And we found that it was difficult to differentiate relapse from reinfection within these genomic clusters. So, for example, if you look at the phylogenetic tree for cluster 9, there was a 3-year-old girl who presented with lymph node TB in 2000. She was subsequently exposed to a heavily smeared positive case of TB in 2017 and was diagnosed with lymph node TB at the same cervical site in 2019. And based on the phylogeny there, it's really not possible to tell whether that was a recurrence, whether that was a relapse or a reinfection. So reflecting a limitation of genomics for this purpose when you've got a strain with little diversity circulating in the community. So the genomic clusters belong to diverse lineage 1 and lineage 4 sublineages. There's no historical evidence to suggest that TB was present prior to European colonisation of Australia, and the phylogenetics is consistent with multiple introduction events from Europe and Asia. So, for example, a lineage 1.2.1 cluster belonged to the same clade as fishes from Indonesia, and a lineage 1.2.2 cluster belonged to the same clade as fishes from Indonesia and evacuees from Timor-Leste. So this does suggest possible introduction from Asia. However, when we looked at the temporal signal, the signal was poor within the data, and so we didn't proceed with Bayesian phylogenetic analysis to predict the timing of when these isolates may have diverged. So in conclusion, the TB incidence in top-end Aboriginal people has dropped markedly over the course of our 30-year study, but remains much higher than in Australian-born non-Indigenous people, with most incident cases occurring in remote regions. The mortality was higher and occurred at a younger age among Aboriginal people, and this is likely multifactorial. I would say anecdotally, there are a lot of late presentations and diagnoses, and part of that relates to normalisation of cough in remote communities where there are high rates of smoking, COPD, like chronic obstructive lung disease and bronchiectasis. It relates to staff turnover, and TB may not be on their radar as an uncommon but serious cause of cough. It also relates to access to chest X-ray, which isn't possible in many remote communities and requires a trip to town. Those are some of the factors, and really some of my colleagues here, including Chris and his colleague Professor Anna Ralph, are doing some work looking into some of the reasons for these late presentations. The genomics suggested that reactivation from latency and recent transmission are both contributing to incident TB in the top end. And using whole genome sequencing, we were able to link cases to clusters. And so, you know, I think continuing with contact tracing, but implementing genomic sequencing prospectively could assist with that. I also think we need to do further work on treating latent TB adequately, and Chris Lowbridge is starting a trial of weekly isoniazid and rifapentine as treatment for latent TB. And because this has the potential to be directly observed by remote clinics, it may improve our latent TB completion rates. We found a low rate of relapse, suggesting that our current directly observed therapy program for active TB is effective. And we found that few overseas-born cases belonged to genomic clusters, and that there was no evidence of transmission of drug-resistant TBs. And this suggests that our current practices of screening for active and latent TB among people who were born overseas are effective at containing those cases. So there are a large amount of people involved in this work. I'd like to particularly acknowledge Professor Vicky Krause, who has been working, who is the head of the Northern Territory Centre for Disease Control and has been leading the TB program for many years. I'd like to thank the TB nurses at the TB clinic who have been very helpful in providing contact tracing data and so forth. At Menzies, I'd like to acknowledge the Global and Tropical Health Indigenous Reference Group to whom I presented this work and who gave really valuable feedback, and my supervisors, Professor Anna Ralph and Professor Bart Curry and others at Menzies. At Doherty, Christy Horan has been working on the MDU TB bioinformatics analysis pipeline, and she was really great in sharing her knowledge and access to her analysis sort of workflow. Maria Globen, who was really helpful in pulling the isolates out of the freezer at Vidral for me, and my supervisors at Doherty, Deb Williamson and Tim Stanier, who have been very supportive. And that's it. Thank you.
Thank you very much, Ella. I'm not quite sure how we should moderate question time. Maybe if people want to turn their video on and wave, or else like use the little hands to, okay, maybe starting with Ben.
Hi, Ella, just to congratulate you. It's wonderful to see this work coming out of the Northern Territory, and it's a remarkable treasure trove to have 30 years of data and to be able to sequence that with essentially good news around very low rates of relapse and also very little evidence of transmission from imported strains. So that's wonderful. I just wanted to ask the question about just the origin of Lineage 1 strains in Australia. It's always been suggested that if TB was present pre-colonization in Australia, that would have been Lineage 1. I couldn't see whether there was an over-representation of Lineage 1 strains among Aboriginal patients, and whether you saw any Lineage 1 strains that were deeply separated, I suppose, from more recently introduced strains found in the Indonesian fishermen and people from East Timor. Just it would be interesting to hear your thoughts about the distribution of Lineage 1 strains. And
so the most common lineage among clusters in remote communities was Lineage 4. But there were several clusters which belonged to Lineage 1. And we also had some Lineage 1 strains among fishers from Indonesia and evacuees from Timor-Leste. These were not closely related to the other publicly available Lineage 1 isolates, but they were separated maybe by several hundred SNPs, I think, from memory. Look, it gives an indication to me, in my mind, it gives an indication that those strains have come from Asia at some point in the past, but because of the poor temporal signal within the data, we didn't do the molecular dating analysis to try and get to the bottom of that. I would say I did go back to some of the early sort of descriptions of the health of Aboriginal people in the top end following colonisation, and there was really nothing in there to suggest that TB was existing in those populations on arrival by Europeans. I would also say the Macassans have been visiting the Northern Territory coastline from South Sulawesi for I think around they were visiting from around 400 years ago. And so I think if TB had been introduced prior to European colonisation, that is a mode by which it may have occurred. But look, I think it's hard to know.
We have a few questions that are in the chat, but I think first up we had Norel Sherry raising their hand to ask a question. If you'd like to go ahead.
Sorry, that was just a clap for Ella. I thought her presentation was great.
Oh, thanks, Norel.
In that case, the next question from the chat, Warwick wanted to ask whether there's any evidence of an increased post-treatment mortality in TB patients compared to like the rest of the population. I think that would mean the rest of the Australian population.
So that's increased mortality after you've actually been cured of your TB. That's not something I've looked at. I don't know.
I guess Warwick here. I guess that Greg Fox observed this in Vietnam.
Mm-hmm.
And there's evidence elsewhere, particularly if diagnosis is late, that post-TB lung disease can be problematic and can actually lead to adverse events. But there's also a suggestion that there may be adverse vascular events as well because of inflammatory changes. So
that's not something we've investigated in the population here. I would say as a result of the late presentations, it's not uncommon for people to be left with sort of large cavities or bullae after they've completed their TB treatment. And I'm sure that does increase the risk of further lung infections and so forth. Would definitely be worth looking at in our population, I think.
So there's a question from Ellen Donnan. Because the duration of your trials were quite long, they were asking whether mortality treatment for First Nations people has changed over that study period. So is it still the 12.5%?
Look, I haven't actually looked at that change in mortality, at whether there's been a change in mortality rate over time. I would say that deaths from TB do continue to occur. I can think of several since I've been in the NT for the last 5 years or so. So it remains a problem. Aboriginal people still end up in ICU with TB and occasionally die from TB in the Northern Territory top end.
Would that be related to continued hospital avoidance? Is that like a late detection?
I think it's related to some of the factors I mentioned briefly earlier. So late diagnoses. In some instances, that may be the patient presenting late. It may be related to normalisation of cough in the setting of cough being so common with very high rates of smoking, COPD and bronchiectasis. It may be related to healthcare staff awareness of looking for TB. It may be related to access to chest X-ray and people being reluctant to come to town for that. I think there are a variety of different reasons. And as I mentioned, Chris Lowbridge, who is a postdoc here at Menzies, Anna, he and Anna Ralph doing some qualitative research on some of those cases and what may have contributed to the late presentations. I think it'll be really informative.
Go ahead, Ellen.
Thanks, and thanks, Ella. Yeah, I just think it would be really interesting just to know whether that mortality's changed. Like, there's obviously been a significant reduction in cases over the same period of time, but that and the work that Anna and Chris are doing, I think, would be really interesting when we have a look from an NTAC perspective at TB and First Nations people. So yeah, if you have the data, it would be interesting to know.
Mm-hmm. Yeah, I can talk to Vicky Krause about that. Yeah. Maria?
Hi, Ella. Yeah, it's nice to see it all in print. It was a lot of hard work, wasn't it? It was a big slog, so well done. I just was wondering what your thoughts are, because obviously the rates of TB have gone down in the First Nations people. Why do you think that is? Because when I first started in TB, they were really, really high, and they've definitely come way, way down. And we don't nearly get as many from the Northern Territory as what we used to. So what, and I'm sorry if I missed it, I had to duck out. So sorry if I'm repeating something. What do you think's caused that?
Yeah. So the TB program was actually disbanded in 1982. And following that, the rates of TB rose a lot. And by 1989, I don't have the figure off the top of my head, but the incidence of TB in top-end Aboriginal people was incredibly high. And that was around the same time that Vicky Krause moved to the Northern Territory and joined others at the NTCDC, and the TB program was reinstated with a focus on contact tracing, on targeted screening, at times with chest X-ray in communities, with directly observed therapy, with standardised regimens, and a number of the sort of the core components of public health control of TB that are still in place today. So all of that was reinstated in 1989, and I think since that time there's been a steady reduction in cases. There's quite a bit of fluctuation over time, and there have certainly been a number of what would probably be called outbreaks of TB in various communities at various time points. That, you know, the TB clinic staff, the nursing staff, and Vicky all remember very well. We've had a spike in cases in one particular community this year, in fact, which we sort of need to look at further. Yep. Ben?
Thanks a lot. Just if no one else has a question, I'll ask a second one. I wanted to ask whether you were able to consider specific communities at risk, and whether there were particular hotspot communities where active case finding interventions or so would make sense. Was this your sense that cases were equally distributed in remote Northern Territory communities, or whether there were certain communities where infection rates were far higher than others? And your the trouble of differentiating relapse and reinfection in some of those cases with recurrence, I think illustrates how in some households and some communities the same strain can linger for a very long time. Just your sense.
Yeah, no, absolutely. It's certainly not evenly distributed across the top end. There are certain regions that have much far more cases than others. I think I mentioned that, so the West Arnhem region and the Roper Gulf regions particularly, and there are particular communities within those regions that have been affected. In some of those communities, it's not just one dominant strain. For example, there's one community which has a number of different language groups, and people sort of travel to and from there from a number of different regions, and there are like quite a few different sort of circulating strains that pop up intermittently in that particular community, whereas other communities tend to have like a particular dominant strain. You're quite right that the same people may be exposed recurrently over many years to isolates belonging to the same cluster, and that's, you know, that's why differentiating the relapse from reinfection is so difficult among those cases. Does that answer? And yes, I think there is a role for targeted public health intervention in those regions. That's really the area of expertise of the NTCDC. They do community-wide screening if there are a certain number of cases within a certain amount of time, but that's obviously quite labour intensive, but is something that they do in some instances.
Maybe just to mention, some of them, I think Chris is, and Anna might be well aware of this, but some of the mobile X-ray technology and AI reading options may make the screening of isolated communities far more pragmatic in future, and would be an interesting thing to explore in those highly problematic communities where proactive case finding may be valuable.
Yeah. No, thanks.
So Michaela has asked a question in the chat asking how cases are detected. what's the routine screening in the target populations? And then I had a follow-on, possibly red herring question.
Yeah, so cases can be detected via a number of different ways. In some instances, it's because the patient's presented with symptoms and or signs concerning for TB infection, and that gets investigated with chest X-ray and sputum, and in some instances lymph node biopsy, for example, and that's how the case is found. It does require the clinician thinking of TB and requesting the mycobacteriology, AFB smear and culture on those clinical samples. Other cases are found through contact tracing. So when there's a case of active TB, then NTCDC has a public health response to every notified TB case, and that includes like screening of all the household contacts and so forth for symptoms, and doing MAN2s, and in a lot of instances chest X-rays as well to find cases.
Thanks, Ella. You did mention that there was some community screening in areas where there were quite a few cases that popped up by passive detection. I'm curious, in the very isolated communities, particularly if there, as you say, there might be some kind of people used to coughing because of smoking and things like that. Have there been any efforts to do active case finding in communities that haven't had any cases but are quite remote, just on the assumption that there may be people may not be presenting passively if they don't have if they haven't had cases in their communities?
Not that I'm aware of. So generally that type of community screening would only be done if a certain number of active TB cases have been diagnosed within a community within a certain time period. So they wouldn't sort of do that generally if there hadn't been recent cases in a particular community. No. Mm-hmm.
Thank you.
No worries.
My question, which was following the question about how routine screening works and if it's happening for target populations, I wanted to know what your thoughts were about involving genetic screening at that level. So at the routine diagnostic level, this is coming back a bit to my work, which, like I said earlier, was malaria, but given that there is such a predominance of drug resistance with malaria and there's genetic screening for drug resistance markers, we were looking at MinION, you know, like Oxford Nanopore long read portable sequencing tech. It's just like it's a really cool toy, and there's a bit of controversy about whether it's really the most appropriate thing given the places where malaria tends to be, but it is very cool science, and whether that might have a role in moving away from a research lab to point of care molecular epidemiology. So screening for markers of the parasite or the bacteria, whatever you're looking for, but at the same time, maybe getting some evolutionary epidemiology information to trace the phylogeny, the lineage of where this is coming from, getting some information about drug resistance, strain typing. Do you think that there's a role for that? And you're describing case detection in really remote areas, and now that there are starting to be, though the margin for error and accuracy is still kind of not clinically appropriate, but there's starting to be these sorts of molecular technologies that can work in remote settings outside of research labs. Like, what are your thoughts to moving molecular epidemiology towards clinical point of care?
Yeah.
Go.
Good question. I think there's definitely a role for incorporation of TB genomics into public health, not having it just as part of research, but actually, as I sort of mentioned in the first part of my talk, prospectively sequencing all culture confirmed an M. tuberculosis isolate from all culture confirmed cases, and using that information for prediction of antimicrobial resistance, and also for linking cases to clusters. As I mentioned, that's already happening internationally, prospectively, and in some Australian jurisdictions, I understand. And I definitely think there's a role for us incorporating that here in the top end. Whole genome sequencing directly from sputum for TB is possible, but not straightforward. And as I understand it, isn't ready for sort of widespread implementation just yet. In terms of whether the MinION at the patient bedside in a remote community is the right approach, I think probably not for us here in the NT, in the sense that it doesn't take long for samples to arrive at our public health reference laboratory at Royal Darwin Hospital, where sort of we can sort of process the samples in a laboratory setting as opposed to in a remote community. So I think not just yet for the MinION in that setting just yet. Does that answer your questions? Which there were a few parts, I think.
I think it was a lot of questions in one very long sentence. It does answer it. The other aspect to the question was, so you talked a bit about phylogeny, and you were talking about like the Snip distance and such. That generally hasn't been what I've looked at for phylogeny. I've looked at like if there's neutrally inherited SNPs so that you can look way back. But I guess in the case of what I've looked at, there's particular lineages that are multi-drug resistant and prone to multi-drug resistance, whereas that's not the case. There's no like organelle genomes for tuberculosis bacteria. But do you think that the level of knowledge that is available combined with like where the technology is at makes things like genetic epidemiology a useful tool right now at the clinical level, rather than just something that supports the research understanding kind of level? So like drug resistance markers is one where like, yeah, that's clinically quite significant, though you'd have to see treatment failure in the Australian sort of setting for you to need to look for it. But if there's other reason why you should do strain typing by way of course in a clinical setting.
Look, I think there is a role for routinely using sequence data for AMR prediction in TB. The reason for that is that TB is a really slow growing organism, and there's often a delay between diagnosis and the availability of phenotypic susceptibility information. So genomic sequencing does have the potential to provide that information earlier than the phenotype data, depending on sort of the stage in the culture workflow that the sequencing is done. So for example, if it could, I think some places are using midget broths and sequencing, extracting DNA and sequencing from those, in which case that information would be available prior to phenotypic susceptibility data. There is also quite good correlation between genotype and phenotype for some drugs, particularly isoniazid, rifampicin and the quinolones. And so that information is very helpful, potentially very helpful to the treating clinician. That said, I'm not an expert in TB AMR, and it wasn't a focus of the PhD work that I was doing. There are probably people on this conference call who may want to pipe up and correct me if I've said something that's wrong. The other part of your question was around phylogenetics and clustering. I think that is potentially helpful for public health. So not necessarily for the treating infectious diseases or respiratory physician, but for the public health unit, in terms of it's an additional bit of information on top of their sort of shoe leather epidemiology, that, you know, the information they obtain from talking to the patient and doing the contact tracing and so forth. So it definitely does have a role in both clinical and public health practice, I think.
I'm biased, but I tend to agree. I love molecular epidemiology. I feel like we need to integrate it as soon as we can. I think we have time for another question. Is there somebody else on the floor who would like to ask something else? I don't want to monopolize this, as I tend to do. Nope.
There's
Nobody else would like to ask a question?
Brooke Berry says she's happy to say something from a Queensland perspective
Yeah, okay.
in the chat.
Hi, Ella. I can't see anyone else from the team. I'm a bit of an outsider, so I'm here looking after some projects, and I'm actually a chemist. So my background is pharmaceuticals and clinical trials and research. But I guess the approach we're taking is, regarding is phylogeny useful? It's as useful, as you say, it's as useful as the data we have. So the sooner we start collecting that data routinely and doing the testing routinely, the sooner we can get to a point where we are confident that that database is robust. At the moment, we've been relying very much on isolated studies that may have population bias or geographical bias. So we're very excited to be coming on board and hopefully contributing to, I guess, that pool of knowledge. And I guess that's the big question is, are we ever going to be able to stop doing lab tests on sensitivities? Maybe, maybe not. But, the sooner we get started, the sooner, and as a routine thing and just have it embedded, the sooner we can answer some of those questions.
Yeah, thank you.
I would also add that the sooner that it becomes routine, the more we contribute to data gathering on a global level. Like you were saying, some of the TB cases are going to be imported because we're in Australia, and they're going to be imported from places that might not be in a position to contribute to, global databases. And so if we're doing that, then it's, we're in a position where we can, and some would argue there is an onus for us to do that.
Absolutely, and also the other way around. Like if we're exporting pathogens, then we want to know what we're sending out.
Yeah, we don't often talk about it in that from that perspective, but it's a good point.
Thank you so much for talking with us today. Do we have one last question somewhere on the floor, or is everybody satisfied? Okay. Well, that's our time. So thank you so much, Ella. I have recorded this and I'm going to post it up onto our website for the other people who are interested. Would you be happy for me to make your email available to anybody who has some questions they want to ask you directly?
Yeah, that's totally fine.
I won't just post it. They can ask me and I'll give it to them. Thank you so much for your talk, and congratulations on all your work. It does look like you've really earned it, the PhD, I mean.
Yeah, thanks so much for the opportunity to speak. It's been a good discussion. Thanks.
Cheers. All right. I'll see you later, everybody.